This is an ion-gated channel. Capsin, acting as a ligand in the reaction, binds to the active site of a TRPV1 protein, which is acting as a ligand-gated ion channel receptor in the reaction. The binding of the capsin to its binding site on the protein results in the channel opening up to let ions in the extracellular fluid enter the cell. The reaction will result in the cell's regulation of temperature.
Epinephrine is released from the adrenal glands that enters the bloodstream when a threat is perceived. Epinephrine then docks to the G protein-coupled receptor on liver cells, which then activates the G protein. Once the G protein binds to the G protein-coupled receptor and GTP binds to the protein, the G protein then binds to adenylyl cyclase. Once they bind, it causes the adenylyl cyclase to change shape, catalyzing the conversion of ATP into cyclic adenosine monophosphate, also known as CAMP. CAMP then signals protein kinase A, which phosphorylates phosphorylase kinase, which in turn phosphorylates glycogen phosphorylase. Once glycogen phosphorylase is active, it cleaves glycogen-releasing glucose monomers, which enters the bloodstream. The glucose provides energy in order to escape from the perceived threat. Intracellular receptor, the hormone aldosterone diffuses through the capillary membrane and binds to the mineralocorticoid receptor in the ductal cell. The hormone receptor complex is activated by the binding and enters the nucleus to bind to the DNA. The DNA is transcribed into mRNA. The mRNA then leaves the nucleus into the cytoplasm to be translated by a ribosome. The ribosome creates more epithelial sodium chloride channels and sodium-potassium pumps. The epithelial sodium chloride channel allows for the diffusion of sodium ions from the lumen to the ductal cell, which then go to the sodium-potassium pump. The sodium-potassium pump allows for the diffusion of sodium outside of the cell and the diffusion of potassium into the cell with the help of ATP. The diffusion of sodium outside of the cell and potassium inside of the cell allows for the regulation of blood pressure. This is an RTK receptor, also known as a receptor tyrosine kinase. An epidermal growth factor molecule acting as a ligand in the reaction binds to the receptor tyrosine kinase protein. Phosphates from the ATP are added to the RTK, which then activates RAS, which then binds to and activates rapidly accelerated fibrosarcoma, also known as RAF. RAF then phosphorylates and activates mitogen activated protein kinase, also known as MEK. Extracellular signal regulated kinase, ERK, is then phosphorylated and activated by MEK. ERK then translocates into the nucleus and phosphorylates c-MYC, activating the genes that promote the cell cycle and growth.